Hello everybody, I am Dr. Farhan Zamir, an adjunct professor and academic specialist at Biotechnica, Bangalore. For a very long time, most of our students who have finished their masters, they are in a constant urge of writing a proposal. And most of them are PhD aspirants, and many a times they ask, sir, what are the interesting topics, or which are the topics of the boom or in the current buzz I can actually start my PhD career with. So we thought of giving you the top 10 PhD life science research ideas so that you can pursue your PhD. Let's try to look into it. Welcome again. So for today, we are trying to understand the top 10 trends in the research area, especially the biological research area, wherein most of the PhD students are aspiring for. So what we will try to do is, we will try to give the top 10 best PhD titles or captions wherein a PhD student can look into it. Let's try to look into it. So what we have done is, I have tried to amalgamate all the branches of biological sciences which are in most of the current buzz. So we thought of classifying it into 5 major domains. The first domain would start with the nanotechnology, and with nanotechnology, I have both nanosciences and nanotechnology as a component. Now, the next one goes with the micro, the next one goes with the phyto, the fourth one is with macro, and the fifth one, I have clubbed it into both preclinical and clinical models. So what we have done is, you can bifurcate, you can look into the entire arena of biological research, wherein it could be either nano, or it could be micro, or phytochemistry, or some macro technology, and then finally going into preclinical and clinical models. Let's start with the first methodology, or the first topic of our interest. So the first topic would be on nanosciences. Now please remember, here I need to make a proper differentiation. The differentiation between nanosciences and nanotechnology. Many a times it has been found that most of the students, they think that both are same, which is not the case. Now, how do I differentiate them? Now, nanosciences are it's a science wherein, or it's in an art, or it's a skill of developing a material, or developing a nanomaterial, to be very precise, at a nanoscale. Now, this can lead into various kinds of approaches, and it could be chemical methods, it could be redox methods, or it could be, the most popular right now is the green synthesis method. Now, with this green synthesis method, people are trying to look into how can I use the technology which comes from either retrosynthesis or from the chemical synthesis, and then develop it towards a green synthesis. And here, from a macromolecule, you are trying to convert into a nanomolecule. And when you do this, as you always know, that at a nanoscale, the properties, that is the physico, chemico, and biological properties of any material changes. Now, if I am trying to start with this kind of a research, then nanosciences is one major avenue. So here, what we talk about in most of the nanosciences, people are trying to have solutions for waste management, to develop newer materials for either for diagnostic purposes or for treatment purposes. And many a times, most of the chemical industries, they have high risk for macromolecules, and the technicians or the workers or the employees are at high risk. So how can I use a green synthesis method so that the technicians are not affected? And very importantly, how can I use nanosciences so that I have a very less or no impact on the environment? And finally, it is the cost. So how do I reduce the cost? Because when I'm converting from a macro to micro to nano, a cost is involved with it. And if I'm able to come up with cost-effective technologies, that's an intention wherein you have a huge impact. You have a huge impact on the society, huge impact on the economics of a country, and very importantly, you can save the environment. So all these three plays a very, very important role in looking at nanosciences at a different level. Now, let us go on to the next part. As I was trying to discuss, nanosciences is the synthesis part. Once I synthesize, what do I do? So for every nanoparticle, if there's always a purpose. Now with this purpose, we are trying to develop technology. Now nanosciences is you synthesize the particle, but nanotechnology is you get the application out of it. So please remember, in nanotechnology, I need to get a technology out of it. I am trying to apply a technology on a nanoparticle so that it would be much more beneficial for human race. So here I could either use these nanoparticles for developing certain manipulative technologies, wherein it can be used for either in a three major ways, to be very precise. The first major way is either it could be used in the formation of a newer therapeutic agents, or this could be also used for, very importantly, the diagnostic purposes, and not to forget, you can still use it for targeting molecules. Okay, so many times the drug delivery system is being done using nanoparticles. Here you can use 4 major or 5 major kinds of nanoparticles. Either you can use a core shell, or you can use a spear, or a triangle, or even a rod or star-shaped molecule, so that you increase the absorptivity, you increase the bioavailability of a molecule at a nano level, so that it can show a better absorption and better action in terms of its mechanism. Now, these are the, again, various methodologies or techniques wherein, you start from drug payload. You can look into the various kinds of X-ray bombardment, wherein rather than me using X-ray, can I still use a nanoparticle so that this nanoparticle will be able to efficiently deliver the drug. And very importantly, as X-ray would have an harmful effect on a human body. So if I am somehow able to, amalgamate a nanoparticle as for a diagnostic or a detection purpose, it would be again a great component of nanotechnology. And very importantly, people are now trying to have something which is called as nanoprobes. That is specially nanofluorescent probes. So nanofluorescent probes are those molecules which can give you the real-time images of a drug and its delivery mechanism in a given host. Now, that was with regard to the nano component. The next component is to understand the micro, and micro talks about the microorganisms. So when I talk about the microorganisms, we need to have, from time immemorial, people are trying to work on bacteria. Especially when I talk about microorganisms, there are 3 major, classes of microorganisms, which are, many a time they are actually friends. But however, the majority of the microorganisms can also have a potential to cause disease. So they are pathogenic in nature. So what people or what scientists are trying to develop on is, can I design certain anti-molecules? Either I can manage this particular microorganism, or I can kill it. Okay, I can bring it into a static condition, or I can actually create a sidal effect using certain mechanisms. But even as we know that bacteria or microorganisms are much more intelligent than us. So what they do is they exist in different forms. So the phenotype is entirely different. So many a times under laboratory condition, whatever bacteria I grow, this is in a formation of a free floating form, and this is called as planktonic form. But however, when it comes into the pathogenesis, the bacteria is able to transform from a planktonic form to a sessile form, which is called as biofilm form. So understanding the various signaling mechanisms into bacterial biofilms plays a very, very important role. And however, again, their management is again a huge issue, because there is a fantastic signaling which goes between the mother bacteria and all the daughter bacteria, and this signal mechanism is called as quorum sensing. This is also one of the major topic of research interest. Now, as I was trying to tell you, that there is a beautiful signaling between the bacteria which tries to reside onto a surface. This is called as an adhesive surface, and once it adheres, it goes on to the next mechanism wherein it will try to create a hexapolymeric substance, and this hexapolymeric substance gives you the bacteria a shelter. Now within this particular shelter, bacteria is trying to have signaling mechanisms, and also it is trying to create a shelter so that even if you are trying to put a bacteriostatic or bactericidal drug, this drug simply does not penetrate into the heap of this particular cell. So the drug in fact comes and then bounces out. So again, we need to look into the various mechanisms here, and then another important molecule, the other important topic what people are trying to dwell upon and try to understand is how exactly your drug molecule, maybe it could be a chemical molecule, or it could be a phytochemical molecule or retrosynthesized molecule, how are these molecules are efficient to kill bacteria when the bacteria is in its biofilm form. So again, there are various kinds of mechanisms which could be elucidated. There are one component system, there are two component system, there are various kinds of master transcriptional regulators which could be studied, and I see this as a very important and a potential field for research. Then, not to forget, once we talk about bacteria, you have the next problematic organism, that is fungus. Now the entire fungi actually creates a major havoc, and as you know that most of the fungus from our day-to-day life activity are being picked up and can lead into such a huge complication wherein we still do not have drugs to actually work on the fungal systems. So the other important component is this fungal can come as a spore. This fungal could be ingested. This fungal during implantation, this can be just translated or this could be transfused through the blood, or many a times this can also go into a nosocomial infection through a catheter infection or whatever might be the source. The problem microbe is the fungus. Now what do I do? So if I'm able to create either one single drug or a combination of antifungal drugs, this would be of a great boon. Now, here there are a lot of existing challenges. As we humans, we evolve, the same thing applies with the bacteria, the same thing applies with the fungus and the viruses. Even they evolve, and there are new systems which are being developed, and hence understanding these systems becomes much more challenging. So newer diagnostic methods has to be developed, and right now, to be very frank, most of the medium or the culture medium or the nutrient medium what we are using are simply not able to support the microorganisms. So there are two major classes. One is culturable microorganisms and the other one is non-culturable microorganisms, and most of them, whatever we understand, is between 35 to 40%. So rest of the 60% of microorganisms, they come under non-culturable microorganisms. So understanding and developing antifungal drug is again a huge interest of research. Now there are already certain antifungal drugs which have been available, but if you look into the entire chemical molecule, most of the antifungal drugs are at the world market, at a global market. You have only 6 to 8 different types of basic structures. Basic chemical structure remains the same. There's only a few molecules which undergoes derivatization. The same thing is being, the same formulation is being sold with various generic brand names. So just make sure designing an antifungal drug would be one of the major contribution to humankind. Not to forget, the next component is the viruses. Now these viruses, with the COVID, we faced a huge pandemic, and with this huge pandemic, most of them, they stayed with us. They became the survivors of COVID, and and few, they had to leave the face of the earth. But however, just a small viral molecule which has only two components. One is the protein component and the other one is the nucleic acid component. Here it might be either a DNA virus or it could be an RNA virus. Here we were not able to actually understand these only two molecular components. So designing antiviral drugs becomes a primordial requirement. And when you look into the SARS-CoV mechanism of how exactly it enters into a particular cell, into the host, and what is the chaos it actually creates. Many a times, with all different kinds of mutation, all kind of spike protein variation, we are simply not able to define and normalize one single antiviral drug, and hence working on an antiviral system can also be very, very interesting. Now, I just finished two components. That was one on nano and the second one was on micro. Now, because of lot of side effects from various allopathic drugs and various other chemical moieties, now there's a huge boom towards Ayurveda and phytochemicals. So phytopharmaceuticals and antioxidants plays a very, very important role, and most of the researchers with various kinds of backgrounds, they are trying to explore phytochemicals for various kinds of bioactivities. So I feel that phytochemical research would be also one of the major interesting area. The next component is there, wherein you have the computers and biology getting amalgamated with each other, wherein there is a concept which is called a drugs design. Now, if you are very good with coding, or if you are good with designing drugs, with chemistry, biology, and a small amount of statistics with bioactivity, wherein whatever you design in terms of your practical, that is in your in silico data, if you are able to validate it using your wet lab data, this becomes a great boon, and we see as drug designing as one of the important component which can lead into various kinds of drug discovery. So normally we have spoken about the pipeline of drug discovery. I would not try to dwell more upon it, but what I'm trying to make you understand is understanding the structure activity relationship studies is one of the major concern, and most of the jobs even for MSc passed out students would many a times has been offered by a lot of pharmaceutical companies. The next important domain is your preclinical data. So once I have something from the in silico, then people want to validate it. Now, how do I validate it? So validation happens at two different levels. One, by having it executed or being tested using preclinical models. And in preclinical models, I have again two models which are available. One is called as the invertebrate model, and the other one is called as vertebrate model. Now, in an invertebrate model, you can use any model. There are certain advantages of invertebrate model, wherein very importantly there are no ethical concerns or there are no ethical permissions which have been required until and unless you are using them in a safe environment. So many a times you can either take up C. elegans, you can take up roundworm, you can take up Drosophila, arthropoda. All these organisms could be utilized. Now, once you look into the in vitro studies, especially you can look into your preclinical studies and then again look into the in vitro analysis of it. But on the other way, you can also go on to a fly model, you can go into a C. elegans model, you can go into a nematode model of it, and then with that translation component, that is from you started with in silico, then you looked upon invertebrate model, and once you look into the invertebrate model, if you want to again validate it, you can still go on to the in vitro digestion studies, and once you know about it, then finally you can still bring in technology and then validate it and develop a technology. And the most happening kind of a technology around AI, ML, and deep learning is with designing the application of microfluidics. You are able to design organ on a chip. So right now people are just trying on organ on a chip. The intention on a long term is to create human on a chip. So you have all the biomarkers from your body, and these biomarkers have been just put onto a chip, and with the help of one drop of blood, you are able to tell what are the pathogenic conditions, what is the body pathology in a given system, and not just with humans. You can look into any kind of a host system, and this is a technology of future. Then not to forget, you have the vertebrate model, and in the vertebrate model you have the rat and mouse, and you have various other systems which could be used. And many a times you can look into the percentage of the various animal models which have been used. It starts with your mice, then you have rat, zebrafish, rabbits, birds, then other animals such as dog, cat, primates are being used. But especially I would suggest you people to look upon these very important organisms, which models which are having a huge amount of data which is already available. Around 72.9% of experiments in India or in most of the biological research laboratories, they work with mouse, and then rat as another translational model, and then very importantly zebrafish, Drosophila. Okay, Drosophila comes under the invertebrate model, but whatever components you study with the invertebrate model could be translated into fish model, that is very importantly the zebrafish model. Then not to forget, most of the immunological research is going on with rabbits. So once everything goes on well, with all the ethical clearances, then you can still work on the primate model. Then once I have the entire preclinical data cleared, then I go on to the major component of the story, which is called as clinical trials. For doing clinical trials, you need to be trained with certain things, and very importantly, clinical trials, especially with cancer research, is becoming a great boon, wherein more and more people are actually required for performing cancer research. Now, clinical trials has 4 major phases, and that is phase 1, phase 2, phase 3, phase 4, and wherein with phase 1, people will just go with the safety and the dosage component, and phase 2, you can increase the patient from around 10 people or volunteers, from 10 volunteers to 100 volunteers, and then look into what are how exactly the mechanistic action of the drug is working. And then finally you can onto the phase 3, you can look into the various kinds of combinational studies. You can still compare your new drug with that of your test drug, and finally you can go into a larger analysis of people, wherein thousands of people could be involved, and so that you are looking for all ADMET, that is absorption, digestion, excretion, no toxicity. If all these things happens, then finally with all this data you go for an FDA approval. So once the drug has been approved by FDA or by the national authorities, and your drug is then available in the market. Now these are the various phases. We just to summarize, we started with the nano, then we looked into micro, then we looked into macro, then we looked into the phyto, and then finally with the preclinical and clinical models, we were able to know that what are the various topics or what are the various fields which a person after MSc or a person can take up for his or her PhD. Now, this was just to give you an idea that what are the happenings or the buzz research which is going on. And any point of time, if you feel that this video was useful and if you want to know something in detail about a particular topic, please let us know. We will be very happy to create more content for you so that you people can get benefited. So all the very best for all the PhD aspirants. So thank you very much for watching the video. In future, if you require any kind of an help, please make sure you write to us on Biotechnica. Thank you very much. All the very best for your PhD. Do well.